All right, what's up? Is this thing on? Okay, so I'm here to make a video here for the free response question on the AP Classroom 2020. This is Unit 4, which is proteins, right? Still, yeah, basically proteins. That's the unit there. So we're gonna look through this question. We're gonna talk through it, and yeah, all right. Give you some general tips along the way, but more specific to this type of question. Okay, so first thing when you look at a free response question, you want to be looking at whatever is easiest, whatever comes to your eye first, okay? So for me as visuals, it's gonna be the graph. I like data, I like graph, I like visuals, so I'm gonna look at that. I'm gonna look at it for a couple of minutes, okay? Just to try and get as much information out of this as I can, and then I'm gonna jump to part A, okay? Only after that am I gonna jump back to the rest of it to try and like fill in the details and get the gaps and whatnot, but we're going for speed here, okay? So what am I getting out of this graph? Okay, well, there's two graphs. Basically, we have check the axes, the x-axis time after hormone addition. Okay, so we got time, it's a classic X. And then the y-axis is the level of phosphorylation of STAT5 and the level of phosphorylation of protein kinase B, okay? So protein kinase B reminds me of protein kinase A, which is a signaling pathway kind of a thing. And a protein kinase is like going to be some sort of motion, works with a protein, does something. And then phosphorylation makes me think of ATP, makes me think of powering up a protein, dropping off the third phosphate from ATP to ADP. Just from phosphorylation, I think there's some sort of like powering, some sort of activation, something. I don't know what STAT5 is, but it's being phosphorylated as well, okay? So relative to maximum, the fact that it goes 0 to 100 and it says relative to maximum, that makes me think that like, okay, maybe this is kind of like percentage, okay? We'll look into that a little bit more, but so what I'm seeing is in my key, the growth hormone is the open box, and then insulin is the filled in circle, okay? The dark line. And on the left, STAT5 gets super phosphorylated in the presence of growth hormone, but with insulin, it has no effect, okay? Then on the other side, it's the flip. Graph B shows me that growth hormone leads to almost no phosphorylation of protein kinase B. You should notice that it's not 0, it's like 5, but growth hormone doesn't have a big impact on PKB. Insulin, however, is spiking like a mofo, and it's really high, and it's kind of staying high for 240 minutes, okay? 240 minutes, quick calculation, what is that? It's like 64 hours, okay? Something like that, right? So we've got 0, 30, 60, so check the minutes. We're talking about, you know, half hour, hour, and then it jumps to 4 hours there. It basically says that little lightning bolt in there says like, all right, a lot of time has passed. We're not gonna give you all the data there because it's not as important. Basically, it's gonna be following the same trend line that you see, right? And so they're going from all the way to 4 hours, right? Which to me makes me think we got growth hormone, we got insulin. That to me makes me think of like a meal, like 4 hours, like a meal, like the impact of that stuff I'm talking about, okay? Then the next thing I'm gonna jump to is A, okay? Describe the first interaction that triggers signaling to begin in a target cell. I'm reading this and I'm noticing that it might really not be talking about this specific example. It just says describe the first interaction that triggers signaling to begin in a target cell. Not the target cell, not the hepatocyte that they mention up top, just like a target cell in general. So maybe I can look up and be like, oh, hold on. I'm seeing growth hormone are protein hormones that regulate carbohydrate metabolism, so it makes me think of food and eating, by hepatocytes, liver cells. So liver cells are breaking down carbs, and that's regulated by the protein hormones, insulin and the growth hormone, okay? Through the activation of intracellular signaling pathways. Intracellular means within the cell. Researchers investigated whether two hormones trigger the same or different intracellular signaling pathways in hepatocytes, right? and the researchers added each growth hormone and some parallel cultures of hepatocytes. So we got liver cells that are experiencing growth hormone and insulin, and then we've got and basically in both things we're measuring how much of the STAT5, okay, and then how much of this PKB. It doesn't really say anything about what STAT5 is or what PKB is, okay? So they are kind of relying on your knowledge from chapter 11 that like protein kinase B is like protein kinase A, which is like a protein kinase, which is a signaling protein, okay? STAT5 doesn't mean anything to me. I don't think it should mean anything, okay? So they added either growth hormone or insulin to parallel cultures of hepatocytes at several time points, okay? They purified the intracellular protein from the cells and quantified, measured the number of the phosphorylated forms of the two proteins involved in two different signaling pathways. So I would write the word different down because STAT5 and PKB are two different signaling pathways, because that's the biggest thing. We recognize that this is about signaling pathways. So it says signaling pathways, signaling pathways, signaling pathways 3 times in this question, right? So you should be recognized this is about signaling, okay? This isn't necessarily about making a protein. It's about signaling, activating a protein, okay? All right, so now we got a little more information. So it says, describe the first interaction that triggers signaling to begin in a target cell. I still think that's a really general question that is saying, what's the first thing? Well, you got a phospholipid bilayer, and in that phospholipid bilayer, we have peripheral proteins and we have integral proteins, okay? Peripheral protein, integral protein, okay? And these proteins are sticking here, and when something comes and lands on a given protein, here's a little battery, it lands on that, this is going to change the protein shape, okay? And as soon as this protein changes shape, then it might be connected to something that then releases, okay? So like you could think about it with like this here pepper grinder, right? Okay, and then so this little battery comes and lands on the pepper grinder, and that causes this to turn, and then pepper comes out, right? So that's the basics of signaling, and I think that that's all they're asking for. Now we use some vocab. I want you to use strong vocab every time that you talk about this stuff. So what is this thing that comes and lands on the integral protein? That would be a ligand, okay? A signaling molecule, a ligand. This can be food, this can be protein, this could be an amino acid, this could be the carbohydrate that they mentioned in the question. Oh, a carbohydrate comes to land on a protein, and then that protein changes shape, okay? So you could call that, typically, if we're talking about a PKB, we're probably talking about a G protein coupled receptor, okay, which is when we're talking about GTP. So maybe they're looking for something specific there. So you could put a general statement and a specific statement that says, okay, so we have the ligand that binds to the G protein coupled receptor, and then the G proteins on the other side. GTP is gonna be, well, there's 3 different parts of the G protein that is connected. The G protein is connected to the G protein coupled receptor. And then when the G protein coupled receptor receives the ligand, the G protein is going to break apart, okay? And that involves phosphorylating a portion of the G protein with the phosphate of GTP. And it's going to lead to a cascade of events. I mean, the whole deal with signaling pathways is we're talking about dominoes. You knock down one, you change one thing, it changes the next thing, changes the next thing. The biggest thing, the vocab word you can use there is you're changing the protein's conformation, aka that means shape, right? As soon as you change the shape, it changes the function. It causes it to do something, right? Now, how do you cause that to happen? Well, something has to bond with something else. Now, that's either a ligand or it's going to be a donation of a phosphate, aka phosphorylation, which is what this thing is talking about, okay? So that should take care of A. B, identify dependent variable in the experimental design. Stop right there and just do that. You don't need to read further. The dependent variable is what your data is. Dependent variable is data, okay? So what data did we measure? We measured the level of phosphorylation of protein kinase B and the level of phosphorylation of STAT5. That's it. Write it down. Good. Identify a negative control for the effects of hormone addition. So a negative control is going to be, I looked this up, a control in which no response is expected, okay? So it is one where like they don't have to do the experiment. They're not like unsure. They're like, okay, look, we know this thing is not going to do anything in this situation. And so we have a baseline to compare against. So as we see up there, what's the one that does absolutely nothing? Well, in graph A, STAT5 phosphorylation with the presence of insulin is 0. So how much does insulin impact STAT5? 0. The fact that it's 0 and they collected the data on that, it just kind of makes me think like they knew that that was going to happen, right? Because they know that it's a flip-flop. They don't really clarify that, but it definitely seems like they've collected data on insulin for 4 hours and nothing happened, right? So they probably they probably knew that there was a difference. They just wanted to see how much does growth hormone impact STAT5, how much does insulin impact PKB. Therefore, I would say the negative control is insulin to STAT5, and I think the negative control is also growth hormone to PKB. Okay? Identify a likely reason why the researchers continued their experiments for 240 minutes. As I said before, if we're talking about liver and carbs and processing carbs and eating carbs, we all know what carbs are, right? So you eat them. How often do you eat? Every 4 hours. Unless you're doing intermittent fasting, which is super healthy for you, and you should probably look into that. But, yeah. So, mealtime. Now let's jump to C. Describe the hormone and signaling protein combination that produced the greatest and most prolonged response. Okay, the hormone and signaling protein combo. Okay, all right, so we know what the hormones are. They are insulin and growth hormone. Okay, and the signaling protein combo. We know what signaling proteins are PKB and STAT5. Now, also, this is really interesting, actually. I haven't thought about this before. You should talk to yourself while you're doing this test. This is the only test in the history of AP tests where you are allowed to talk to yourself. Good God, okay? Speak things out. Talking through things verbally is better. If I was sitting here in silence doing this, I would not figure it out as fast. Guaranteed. The fact that I am talking to myself allows the thoughts to go from my brain to my mouth, get processed. They go out, it goes into my ears. Extra processing means faster learning. Guaranteed. The other thing is you should also extend your brain onto a piece of paper. Always have scratch paper ready so you can write words down like different or combo or hormone and signaling or hormone, insulin, growth hormone. I mean, that takes 2 seconds for you to write down, but it's going to extend your brain onto the paper. When we extend our brain outside of our brain and then use sensory input like our eyes or our ears to bring it back into our brain, that cycle is repetition. Repetition is learning. Okay? Use it. Talk to yourself. Okay? Then produce, I'm not just trying to make myself feel better. All right, so the one that produced the greatest and most prolonged response. It's so easy. So the biggest thing here is cite the data. So all you have to say is in graph B, we see that protein kinase B within 10 minutes spiked to over 90% phosphorylation comparatively, relatively. Okay. And then remained above 80% for 4 hours, not over 4 hours. For 4 hours, it remained above 80%. That's all you have to say. Cite the graph, use the vocab, use specific numerical data. Next. For the phosphorylated STAT5 response to reach 100% at 10 minutes in figure 1A, calculate, you see how they follow that format. That statement right there is exactly how you should be answering questions. For the phosphorylated STAT5 response to reach 100% at 10 minutes in figure 1A, that is how you should answer things because that's how they're phrasing things and it shows that you're looking at the things correctly. Okay, so it says calculate the rate of decrease, percent decrease per minute and the detected level of STAT5 from 10 to 30. Okay, so this is where we're going to have some math and this is where I'm going to have to write some stuff down. Okay, so from 10 to 30, it does go from 100 to roughly 35. So it goes from 130 to 35 in 20 minutes, okay? So there's a fancy way to do the math here, right? Like, time equals rate over whatever, but I'm looking at this going like, okay, well, it's moving 65% in 20 minutes. So what did it move in 10 minutes? 30.25. What did it move in 5 minutes? that's 15.125, okay? And then it moves about 3% every minute, right? Because, you divide 5 by 5 and you divide 15 by 5 and you get 3%, okay? I'm not a math teacher. I'm sorry I can't really help you with the ratio on that. Maybe you could, I don't know where the X is on that, on how to calculate the percent rate per minute, okay? So like the rate of decrease, you could call that RD, okay, per minute. So we need the rate of decrease times the number of minutes, which is 20. We're using this sentence and creating an equation from it. So percent decrease per minute, per means times, okay? So the rate of decrease, which I've labeled RD, times 20, because it's 20 minutes over the course, is equal to 65, okay? Because we have moved 65 percentage points within 20 minutes, okay? So I think that is your thing. It's basically X times 20 equals 65, okay? Divide that 20 on both sides, so X is equal to 65 over 20. Guess what that's gonna give you? A little over 3%, okay? There's multiple ways to calculate this. Use your best, right? And then slap it down right there, okay? Remember that you can take pictures of your work. I believe that's how that's gonna work. And yeah, okay? Then we move to D. The researchers claim that the similar regulation, now remember when we get to part D, the last part, they're always gonna ask you some like theoretical question, okay? The first question's knowledge, A, okay? B and C, the middle ones are always gonna be skills. Knowledge, then skills, then they're gonna go theory. The researchers claim that the similar regulation of carbohydrate hepatocytes while the cells are treated with growth hormone or insulin results from the activation of different signaling pathways. Again, signaling freaking pathways, right? So like if you're getting stuck on this and you're just like, oh my gosh, my mind's like, zoom out and pay attention to what they're asking. What are they asking? They're asking about signaling pathways. They're talking about digestion of food, okay? They're talking about hormones. So jog your brain of like, what does it mean? Phosphorylation, signaling pathways, hormones, right? This is the chapter we didn't get a chance to go over, but you should still have a basic concept of what protein activation looks like, what proteins do, and how molecules interact with them, okay? So the first one's a statement. It says, use the data from the researchers' experiment to support their claim. I mean, they said use the data. So again, you are going to reference the data as specifically as they did in part C, where it says, reach 100% at 10 minutes in figure 1A, okay? You need to use that very specific language coming directly from the thing. So I'm gonna say they're asking about, they claim that the similar regulation of carbohydrate habit by hepatocytes when cells are treated with growth hormone results from activation of different signaling pathways. So their key thing is here is different signaling pathways. That was in the beginning too. Remember when I said different? Okay, so different signaling pathways. They are claiming that this comes from different signaling pathways. Now, the only way that two different hormones would activate two different signaling pathways, the only way we could prove that is if we did a test and we said, all right, let's put these two hormones into a cell and check the downstream effects. Let's put these two hormones into a cell, check the downstream effects. The downstream effects here are PKB and STAT5. As you see in figure 1A, insulin has no effect on the phosphorylation of STAT5. However, in figure B, insulin has a huge impact, don't say huge, say insulin jumps to a 100% phosphorylation of protein kinase B within 30 minutes and remains above 80% for the remaining 3.5 hours. However, growth hormone only leads to a 5% phosphorylation of protein kinase B for the entire 4 hours. Therefore, it seems as though insulin is interacting and activating protein kinase B signaling pathway, whereas growth hormone is activating the STAT5 signaling pathway by phosphorylating it within the first 10 minutes at 100% and then remaining above 20% for the remainder of the 4 hours. That's all you got to say. Okay? The graphs show that growth hormone is impacting STAT5 and not protein kinase B. Insulin is the opposite. Okay? Now don't say things like insulin is the opposite. I'm just saying that because I'm repeating myself, okay? State these things out very clearly. You can't assume that they understand what you're thinking at all. You have to write out every single specific detail, okay? Don't say words like it or that, okay? You got to say specific words, not like the hormone. Say growth hormone, say insulin. Don't say the protein. Don't say the graph, okay? Say figure 1A, figure 1B, protein kind of, use the specific words so they know exactly what you are talking about. And then we can prove that you know what you're talking about, okay? Nice job. Get as many points as possible. And remember, you don't have to get all the points to get a 5.